here today from the University of Maryland. I'll just say a few words and then get out of the way so Dan can give his presentation. He got his BA from Oberlin and a PhD in physics from UC Berkeley. So we both tread on that campus, I guess roughly, we only missed each other by 3 or 4 years. He works primarily in the area of computational theoretical neuroscience, and his work is characterized by notable collaborations with a number of neuroscientists who are running wet labs, making use of cutting edge tools in statistics and machine learning. But what I really appreciate about a lot of Dan's work is sort of the clarity and the detail with which he unpacks the problems he's concerned with. He's done some really beautiful work on neural systems identification, developing rather sophisticated procedures for subunit model fitting. He's got a recent interest in cross-species comparisons of the role to which movement can modulate neural activity in the early visual cortices. He's done some very nice work recently on the detailed characterization of neural selectivity in freely viewing primates. But today, I think he's going to tell us about some work on the neural basis of disparity selectivity, which is really the topic that first made me aware of Dan and his work. A lot of some of the stuff you've done with in collaboration with Bruce Cumming on the topic. So that's probably enough for right now. I'll stop talking and let Dan take over.
Thanks, Johannes. In fact, gosh, I feel like giving an intro too, but it's so long that since I am going to be mentioning disparity, I kind of feel Johannes is one of the experts, and so I was a little uncomfortable to do that. And but I'm really glad to be here. I shouldn't even say but. It's great to be here, and I also I do everything more or less, as Johannes mentioned, with collaborators, and in fact. One of my closest collaborators and some of my more exciting stuff is that I have with him. Bevel Conway is coming in 2 weeks, so I decided to actually take a pivot and talk about this disparity problem. And I'm really curious what you think. As you'll see, it loops in some really big picture things. I think that relate to any problem, and then maybe is also interesting from how do you combine information from 2 eyes perspective. So this understandable or inscrutable is kind of one of the large questions we're faced with, I think, as we go forward with increasing sophistication of tools, both computational and experimental. And the specific problem you can kind of view as a toy problem, kind of a classic problem, or I think it's pretty interesting in its own right. So I'll try to bring that along. So, I need to select there. All right. So it starts, I just wanted to start with this quote: "What I cannot build, I do not understand." This is a quote from an old physicist. I was sort of raised as a physicist and thought this was the metric for which understanding came from. And I think it's, yeah, I used to give talks about understanding the visual system for the perspective of then being able to tell computers how to do it, for example. Unfortunately, machine learning has gotten ahead of us, and now this is a picture from a more recent paper of one of the early successful object recognition machine learning algorithms that can take an image and classify it. They've gotten ahead of it, and we can now build these things increasingly complex. They can do increasingly complex tasks, but unfortunately, it hasn't. While it's a step in the right direction, it hasn't brought us a lot closer to understanding. And although it does, in some sense, present a lot of the same questions we've been asking as neuroscientists, like you can imagine in a deep artificial neural network, trying to understand what the contributing parts of it are and what that says about the whole. But the problem is this has been almost a morass in terms of understanding, and really, what it gets to is kind of this going to Mars kind of goals, in the sense of trying to understand really the brain in the largest perspectives, which would be from sort of computation, what the brain is trying to accomplish, algorithm, the specific ways it accomplishes that, and then implementation, the mechanisms. And ideally, when you're studying the brain, you want to understand the mechanisms so you can fix things or figure out the specific roles that contribute to ultimately brain function. And I think we as scientists, and maybe I certainly as scientists, have always thought of an understanding to ultimately relate to an algorithmic description of what the brain is doing and how it's doing it. And the disconnect is now because the sort of data-driven discovery using sufficiently general algorithms does not really relate to algorithm at all. It's almost, we don't know the algorithm, so we're going to give a sufficiently complex network a lot of examples, and it's going to figure out how to do it. And whether it figures out an algorithm or anything is an open question in a way. And ultimately, if that's how the brain works, being able to build something, understanding the mechanism is hard if you don't know the algorithm. And so, as I mentioned, this is meant to be the sort of big wrapper on this, to dive into first the visual system and then primary visual cortex. And one of the ironies is, I think our hopes for the algorithmic view of understanding the visual system came from the same sort of inspiration that led to machine learning and deep neural networks. And at least as I've seen it, it came from this upside down picture. I turned it upside down so it can be oriented of the wiring of neurons in primary visual cortex, which are selective, at least back in the day, to oriented bars, and how this is assembled from getting feed-forward connections. This was a hypothesis at the time, from neurons which project to V1. And this is essentially a mechanism for talking about neural selectivity that starts in the retina, and we understand how these receptive fields, the selectivity of retina and then LGN neurons is constructed from circuits in the retina. This is now a circuit for V1, and the important thing is it's compositional. It shows how you can get selectivity to more complex things from less complex things. And so this was essentially an inspiration from a lot of the early deep neural networks that were, at least some of them were really trying to simulate the visual system. This is a picture that I saw a lot in the 2000s from Tommaso Poggio's lab. And the idea is that if you do these transformations, you can see how the circuit transforms to increasingly complex features, with the features at the top being object recognition, being able to recognize the components of our world. This whole idea of thinking about visual function as feature detectors is pervasive, but somewhat true. This is just a figure from the Kendall textbook where you can see how you have sort of the basic circuit elements go in, and then V1 pulls out and is selective to different types of visual features, color, depth, direction, orientation, complex form isn't there, and that this V1 kind of assembles these and packages them on. And there's this question, is this a good way to understand V1? And the answer I have is, in this talk, is sort of that I'm going to present a dichotomy. On the one sense, I'll show you, and I'll spend most of the time because I'm kind of excited about it, kind of an algorithmic description of how some of V1 works. And this is the kind of thing I think we were hoping for in general for a description of, say, how the visual system works. At the same time, I'll also show you that a lot of the neurons in V1, at least from the experiments that we have so far, are possibly doing something else that is not necessarily algorithmic, but has function on a population level and doesn't really clearly relate to the composition. And that you can have both true in V1, and this raises big questions for kind of how to study it and how to interpret the information. So all very abstract. I'll dive into talking about now the question of binocular integration in V1. So V1 is a great place to study questions like this, especially as a toy system. And to the degree you care about it, it can be quite a sophisticated toy. But you have two eyes that see the image of the world, and the left visual field projects to the right hemisphere and vice versa. And what that means is because both retina see both the left and right side because we're binocular animals, it means there's a crossover, both eyes projecting back to the right hemisphere and the left hemisphere aligned such that they are looking at roughly the same points in visual space. This combination happens in V1. And so the LGN is eye segregated, meaning the same points in visual space are represented along this way, but the different eye information is kept separate. And then neurons in V1 are binocular. And so this is a kind of rudimentary question, is how do binocular V1 cells actually integrate the information from both eyes? And keep in mind that both eyes are looking at the same object, so it's something you would want to do. And with that maybe long wrapper, I kind of now want to go, these look like a lot of points. Hopefully each one is relatively quick, but to see where you're going, I'm going to first of all talk about just some what we know about binocular V1. And then I will show you this, essentially two main parts of the talk. I'll show you the part of my work that argues for an algorithmic explanation for binocular disparity tuning. And that's the sort of, yes, we can take some of V1 neurons and talk about features, and there's some interesting, hopefully new insights for how V1 combines eye information there. Well, what it leaves is a lot of cells that don't follow this rule. And I give another way to sort of understand what they're doing, which is through nonlinear processing and nonlinear mixing of eye information, which is another big theme. And then I'll conclude with what it all means. So I will start with talking about just the basics and the really classical basics of binocular disparity. So what binocular disparity refers to is the fact that that the two eyes see a slightly different version of the world, but you always, when you're looking at something, verge to a point which is somewhere in depth. And that means that if you look at another point an equal distance away, that the image on both retina will move the same amount in the same direction. However, we live in a 3D world, which means some things might be closer or farther away. If something's closer, the interesting thing is that the image moves one way in one eye and the other way in the other eye, such that if you kind of think of the center point where you're verging, they move in opposite direction and have a disparity or a difference in the location of the images in each eye. What this is showing, let's see if I can undrown myself out for a second. It's showing an early experiment that's showing V1 is sensitive to depth. And in this case, this is a ruler driving a V1 cell, which is importantly selected to a vertical orientation. And then as the ruler is moving closer, the relative position on both eyes is changing. And the eyes are only responding or responding optimally when you have the right feature, a bar, moving at the right distance, which then aligns to the preference at a certain disparity for this V1 cell. So it's sensitive to a particular combination of the images in both eyes, which is the left and right eye. So this kind of raises a question. In fact, it's much easier to think about vision as a 2D problem, in part because even if you hold your eyes fixed, the things that you're looking at won't necessarily be fixed. And if you think of us from a circuitry perspective, again, if you're at one vergence and you move something closer, there's going to be a binocular disparity. If you want to actually know have circuits process this image, you actually have to somehow loop it back together such that you're either routing information such that the cells communicating about the blue and the cells communicating about the red somehow push this information back together to account for the binocular disparity. Or you might want to gate the response, like we might have just seen, the response of these neurons so that the cells will only respond to the image at a particular binocular disparity. And how this is solved has been a question of great study, which is how do you combine information for both eyes without knowing their disparity, which you don't necessarily know in V1. So the standard models, which were derived from some wonderful neurophysiology back in the 80s using drifting gratings, is called the binocular, studied binocular simple cells. And the basic idea is we leverage the known filter selectivity of V1 and then have a neuron in V1 that integrates from the left and right eye. And so it does a filter integration of the same filter in both eyes, but those filters can be shifted by the preferred disparity. This is a fairly simple model, and what it gives you is that you'll fire more if you have the right image in the left eye, you'll fire more if you have the right image in the right eye, or the correct image in the right eye, but you'll fire the most if they're in both eyes. And if you start shifting, you'll have things that can be misaligned because there's a sum. And so this is a simple model that gives binocular disparity selectivity to binocular simple cells, the data, which was recorded in gratings quite nicely. And they then generalized this to the more complicated set of cells in V1. And this essentially involves integrating over simple cells from the 4 quadrants, so that you have what's known as phase invariance, or invariance to the exact position, spatial invariance to the exact position of the feature. And in this case, the feature is disparity. And then so you have a phase invariance representation of disparity. By the way, I see there's a chat, and if anyone, I'm hoping I'm hitting the right, right level, but please feel free to interrupt with any questions. So a key point of the binocular energy model is that you have to actually sum linearly between the eyes, which involves a linear integration before summation to a simple cell. Then you have a nonlinearity before it goes to a complex cell. And to get phase invariance, a standard way to summarize this is a squaring operation using the 2 phases. And what squaring does, where you have a linear addition of filtering in the left eye, linear addition of filtering in the right eye, and squaring, what that essentially does is it generates a term that just has to do with the input to the left eye, and then a second term which just depends on the right eye, and then you have a term that depends on their interaction, which is the left eye match times the right eye match. And so that is the canonical binocular energy model, and that's how it works. However, it was pointed out that as soon as you move away from gratings, the binocular energy runs into trouble with explaining real neural responses. And this can be demonstrated as a grating. If I actually multiply by -1, this is actually equivalent to just moving the grating. And this sort of works. Multiplying is just like shifting a phase. And what the binocular energy model predicts is it would only change this last term, but change nothing else. So you have the same response, but if you're correlated, which is to say you have the same image at the left and right eye at the preferred disparity, then you go up. And if you're anti-correlated, which means you have opposite images in the left and right eye, you go down. And that's just what you would predict from the binocular energy model using gratings. as soon as you step away from gratings, this ambiguity between phase shifting and object shifting goes away. And this was demonstrated with relatively simple stimuli, but basically where all you're doing is you're presenting dots to the left and right eye. And you can control, by presenting them different images to each eye, you can control their relative depth. And in this case, if you look closely, you'll see it's the exact same pattern in the left and right eye. And this is a correlated thing and results in a correlated disparity, which is just a shift in difference. You can also multiply the image by -1 and present an anti-correlated disparity. And the important thing is this isn't the same as a phase shift, there's no point in real life where you would actually see an anti-correlated image. In other words, the same image in one eye inverted. It would turn white to black and black to white. It would look very strange. And maybe not coincidentally, when you look at neurons that respond to how they respond to anti-correlated disparity, they can respond positively and negatively, but they respond much more strongly to the preferred disparity and do not have that same negative component with the unpreferred disparity. And this turns out not to just be explained by the fact that this looks like it's hitting the x-axis, because there's also disparity selectivity that's asymmetric, where the preferred disparity has a much stronger response. I'm sorry, the correlated disparity has a much stronger response to the response to essentially non-real images. And of course, this is what the binocular energy model would predict this, but somehow the real neurons are not explaining response to anti-correlated images. So this speaks to two things being important. First of all, that when you go to more natural situations, binocular energy model fails. And I'm sorry, I lost the second thing. What I'll say though is this is still a handcrafted, I've been calling these handcrafted solutions because these are still things that could be presented fairly simply, and you can build a simple model to explain, to build the disparity selectivity. So there's been a host of models to try to come up with reasons that the preferred disparity is selected for, and then the anti-correlated preferred disparity is attenuated. And some of these models have been refuted by later models, but for the most part, I just want to point out that no models could be really directly fit to the responses themselves. And so the true tests of the models would be to go to a data-driven framework, which is what I'll do. And I just partially because of, I'm obligated because of my host, but I also wanted to say that it's also well recognized that studying disparity selectivity in more natural stimuli lead to different sorts of responses than what the binocular energy model could bring. And there's been a lot of good normative work suggesting properties of the filters and different properties of disparity processing and cellular processes that are involved. And in some sense, from that perspective, you could view a data-driven approach as trying to both validate and seeing how they're assembled in single neurons. So that's all the background, and hopefully I'll bring this together now in the sort of data-driven approach. And now I can get concrete and talk about the experiments that that my collaborators done and that we've been working on analyzing. So the experiment is to present a relatively simple stimulus, which is a random bar stimulus, to V1 cells, and it's a flashing random bar. And so these you basically select different positions of black and white images from gray at any given frame. And you're doing that while recording in V1. And the experiment is, the stimulus is modified so that once you know the orientation of the receptive fields, you rotate the bars accordingly. So this gives a simpler stimulus, and one that's sort of, you can always look at the direction of the appropriate disparity. And so what that means is once you have this bar, so you have this particular pattern of bars presented, you've decided that you present it to both eyes using a haploscope. And all a haploscope is it's a set of mirrors oriented so it looks like you're looking forward, but really you're looking on one monitor versus the other. So you look like you're fixating on a point in the distance, but in fact, you're looking at two different monitors, and you can manipulate disparity relatively easy. And what we do is, for a given pattern, it's presented to both eyes, and this, if you look closely, is the same pattern presented to both eyes at the same location. But each update of a new pattern, you also update the disparity so that the next image might be at a different disparity, and so on. And so the disparity is randomly changing in a way that's uncorrelated with pattern. And instead of just being able to move across and measure just a single disparity curve, what you get out of this is a rich disparity tuning curve that's across time lag and across correlated and anti-correlated. And essentially, this is just a spike triggered average, or a forward correlation, however you like to say, but basically looking at the average response to a given disparity averaged over all patterns. And so in this sense, you can measure the same things you measure in gratings, but now you can measure that by averaging the responses across all patterns. And what this is showing is the response to the preferred disparity and the response to the anti-correlated disparity. And I've just inverted the response so you can see the degree of attenuation. And so the issue, though, is that when you have these complicated spatiotemporal models, that you need to then fit models to them to see and see what those models show. And this isn't a problem if you just present simple stimuli like gratings or bars. So the classical way to sort of characterize the selectivity of a neuron is to just present every stimulus that you think is the neuron selective for, and then you have its sort of function characterized, or you can decode its response very easily. As soon as you start having a large range of patterns and possibly disparities, you're in a situation with very high dimensional stimuli, where each data point exists in a high dimensional space where you're not only changing, for example, the orientation, or in this case, the pattern at one location of the image, but you're changing it in a lot. And just for example, if you wanted to present visual stimuli are all naturally high dimensional. If you wanted to try to exhaustively understand a neuron's response to, say, cute animals, you could come up with every possible cute animal, and it would be a very sparse representation in the space, and these measurements wouldn't get you anywhere. For example, if someone then came the next day and took another picture of their cute animal, this cute animal looks different than all the others. It could be described in this space, but you didn't do the measurement. The only way you can kind of understand this, of course, is through a mathematically defined model. And from that perspective, the math just tells you how to sort of use the data and then be able to interpolate to unknown points. And just to sort of think about that in a simplistic way is, if you had a sparse representation of the data, you would want to choose a form of the model that you're going to use. For example, the binocular energy model, or what I have here first is a linear model. And then you would need to choose parameters of that linear model to fit the data. So a statistical model is a combination of a mathematical model and parameters that are used to fit the details. If you choose a different mathematical form, you'll have different parameters and a different interpretation of the data. And ultimately, you would validate the model based on set aside points that you didn't use to choose the model. And that might then help you tell what model's better and worse. in the context of the binocular energy model, the binocular energy model is a particular mathematical form. Sorry. And that's with these adding left and right inputs using filters, and then squaring, and then adding. And in this sense, the parameters of the model would be the actual filters themselves, which are spatiotemporal filters. And so those are these right here. And this is what a binocular spatiotemporal filter would look like. It's showing the sort of selectivity to white and black as a function of time lag back of the stimulus that has been presented in the back across both eyes. And because it's a single filter, it's implicit that this is summed between left and right eye. We enhance this binocular energy model in our data-driven approach to add a linear receptive field, so that if it's a binocular simple cell, we can also explain responses. And we also have a spiking nonlinearity for a couple reasons, including the loss function, but it gives more flexibility to the model. This sort of model, which is a standard now cascade model called an LN-LN, linear, nonlinear, linear, nonlinear. And there's a lot of, it's sort of historically been figured out. It's a very good and easy model to fit. What fitting the model involves is you start with an initial guess for your parameters. So again, we're fixing the mathematical form. The initial guess for the parameters might be, I'm just going to guess that something in the middle, but it's random. And for an initial guess, and then as you subsequently fit, it's an image computable model, which means you can take any stimulus, push it through the model for whatever the parameters are, and generate a predicted response given those parameters. And then you compare that predicted response to the observed spike times. You have a loss function, which essentially chooses to predict higher firing rates where there's observed spikes and lower firing rates otherwise. And by then following the directions of the best improvement of the model, you ultimately end up in the fit to data. And of course, what that looks like is a model that can better predict the spike times. So that's the general idea of the data-driven approach. Again, you choose a particular mathematical form and you apply it to data, and you get, when you use that to fit, in this case, a nice complex cell that we recorded in V1, you get exactly what the binocular energy model would predict. So first of all, this is the response of the neuron to correlated and anti-correlated disparities. But you have more or less no linear filters, and then you have quadrature pair filters. And they're quadrature pairs in the sense that, first of all, you can see both of them are disparity selective at the preferred disparity, and that's the difference between the image in the right and left eye. And then these two filters are 90 degrees out of phase, so they span the whole range of possible phases. So in some sense, this data-driven approach validates the fact that the best description of the data given the mathematical form. However, when you compare the predicted disparity tuning curve to the actual disparity tuning curve, you see that the BEM does indeed have this symmetry between positive and negative, and doesn't predict the responses. Sorry, the magnitude of the disparity tuning curves. This you can do across all the neurons we recorded from. And along this x-axis is a measure of how strongly tuned the disparity of the, how strongly tuned to disparity the neuron is. And you can see that this is where essentially you have noise-free estimates of the disparity tuning. Oh, I'm sorry, this is the predictive power, how well it explains overall, and the disparity tuning. The binocular energy model explains some fraction of the response to disparity, but it's still a relatively small fraction. And so you can see that it also doesn't seem to do well with disparity tuned neurons any more than it would with non-disparity tuned neurons. And so this is now just revisiting the basic conundrum in a data-driven framework is, do you need a specialized disparity? Do you need specialized mechanisms different than the binocular energy model? Or is this just model, if you just fit everything better, you have a more flexible model, whether it would use the same mechanisms and just fit? So that's what I want to address now with this new model, binocular convolution model, and the sort of, oh yeah, question.
So some of these subunit models for fitting neural data explicitly enforce filter orthogonality. So one way to interpret that is the enforce in the fitting procedure that what filters are required. Is that built into this particular approach or not?
Yeah, no, I should have mentioned that this quadrature pair happens naturally, and it happens because there's two filters. If you put more filters in, it might try to squeeze more in, but it doesn't. The fits don't get any better. So yeah, there's no constraints. Spike trigger covariance automatically finds all orthogonal filters, but this doesn't. So you can get so in some sense, these quadrature filters come because it's trying to get the phase invariance, and it needs to do that using orthogonality. The sort of alternative approach when machine learning approaches were coming online was to say, well, let's just be agnostic to the functional form. Let's just fit a deep neural network and see if it works. And I'll just summarize this in a couple of slides. This is, so I'm not going to go into this model other than to say it works, but just so you know, what you can do is you basically can have a multi-layer network that then can be this thought of as a universal function approximator. And it will, the first layer will be like binocular spatiotemporal processing. And note that it doesn't have to necessarily linearly add left and right eye. And then it can have intermediate layers. And then ultimately it predicts the output by integrating over the previous layers. When you do this, there's a couple details. And one of the main ones is that they're convolutional, and also that to leverage the amount of data that you have, you fit them to multiple neurons at once. These are all details that aren't important for this story. The sort of what is important to this story is essentially that when you do this, you can take the same data, the same neurons, and you can really almost perfectly predict their disparity tuning curves. And I was super happy when I saw this. I was like, great, we've solved disparity tuning of V1. And my collaborator, Bruce, was like, okay, so, how does it compute disparity? And that's exactly the problem of deep neural networks. And this is actually especially hard to answer, in part because of two main factors. One is that when you make a bigger network, it will then use more weights. And it's not looking for a specific problem, a specific solution hidden as a small solution hidden in this large network. It will just expand to fill. And what this is showing is just how connected this neuron is to all the units in the previous layer. So it just uses all the space. And secondly, you can make any assumptions. You can start putting mathematical assumptions. And as long as the network has enough components, it will do a pretty good job with some small exceptions. One of the most important aspects of, so we went through this huge exercise in trying to find a way to reduce a machine learning network that worked into a model that we could take home, take to the bank. And so we used one of the most important elements is a convolutional DNN. And what a convolution is, it's essentially reproducing the observation that the visual system sees, has the same processing at each spatial position. And then what it does is that you fit one filter, but that filter is applied across all space. And it's thought that this is actually an on-the-ground model for a complex cell, where you just fit the same processing at different spatial positions. It gives you filter invariance. we maintain that, and we used a bunch of other biological constraints to fit the output of the gosh, I'm sorry, to basically reduce this model down to something that was sparse, and it still had these convolutional streaks, which you could see here. But basically, it was then a model that we wanted to further reduce the biological convolutional network. And so this is now the model. I'll just jump to the model and how it works briefly. What we did is we reduced to a small number of filters, both excitatory and inhibitory, and they get convolutionally integrated across space. And what this is showing is the weight to a given filter across space, and it likes these streaks. And this is essentially the spatial position of the receptive field. And then this is the response to each of these filters that it fit. And it reproduces the same, it produces the response to disparity. And it works in essentially all the neurons. And I really should have sped up a lot. So I'll just say that it dramatically outperforms the binocular energy model. And I'll say how it works. And how it works has to do with convolutions. And the fact is the way a convolution works is, again, it takes a single filter, in this case, a binocular filter, and it shifts them in tandem across both eyes. So if you think of these presenting a stimulus, each version of the filter, shifted version of the filter, is seeing a different stimulus because it's shifted relative to the stimulus, to the same stimulus. Another way to show that is to actually, from the filter's perspective, show what the stimulus being presented. And what you see is you see a collection of stimuli with different patterns. In other words, because it's a particular pattern and you shift it, it's then a different pattern to that filter. Because disparity and pattern are uncorrelated, the response of the neuron is actually summing how much of the response is responding to the pattern versus how much is responding to the disparity. But in this case, the pattern is uncorrelated across the filters. And that means it's kind of like essentially invariant to the pattern, whereas the disparity, each filter is reporting the true disparity. And as a result, the variance of the disparity is summed superlinearly. And so by having a lot of different filters responsive to different patterns, but sensitive to the feature we're interested in, which is the disparity, you end up amplifying the disparity selectivity, and the pattern selectivity gets relatively small. The more filters you have with uncorrelated patterns, the more amplification you get. And for this reason, you get a lot of amplification from convolutional filters. And in that sense, the binocular energy model does do a little better because it has two filters that are uncorrelated with patterns because they're orthogonal, but they are sensitive to the same disparity. So they do get some boosted disparity selectivity, but you can do much more by having lots of filters selected to the same disparity and then integrating across space, which essentially integrates across pattern. And this is kind of the flip side to spatial invariance, which is you're getting spatial invariance while you're also having correlated information from the feature of interest. And so I'll just now just skip to the, not skip, but with that said, I'll just summarize. This is meant to then show you an algorithmic explanation for disparity tuning that is driven by kind of filter selectivity and then putting those together using convolutions. The flip side of this is actually a relatively quick story, which has to do with the fact that most V1 neurons are actually not disparity selective, but they're still binocular. And just to show you that textbook figure again, really only there's monocular, there's thought to be just monocular cells that are relatively few, and most cells are binocular, and most outputs are binocular. And despite that, as you could have seen from previous things, there's only a small set of neurons that are actually specialized for disparity selectivity. And this is really hard to imagine because of what I said at the beginning, which is that you have, if you don't gate by disparity, then how could the response to the two images in both eyes make any sense? But what we did is we went back to these old neurons and looked at not just their disparity tuning, but also their monocular bias index. And what you can see is there's a handful of cells that are monocular, which have, and of course they're not disparity selective. But then as soon as you start getting, you have a big raft of cells that is binocular, but not disparity selective. And this color code, green, yellow, red will sort of convey this. So the disparity selective cells are yellow. The binocular energy model, I'm sorry, the binocular convolutional model I mentioned can simulate these. I didn't mention this, but it both explains the responses of the disparity selective cells and non-selective cells. And what we can then ask is, well, what kinds of solutions does it find for the non-selective cells? How are you not disparity selective? And what we find is a basically unscrutable combination of left and right eye filters. This is now the disparity selective model for one of these cells up here. I'm sorry, the binocular convolutional model. And you can see the excitatory filters. It's binocular because it responds to the left eye or the right eye, but it rectifies. It has a nonlinearity before it does that. And you can see in some cases, there's binocular subunits in inhibition, but almost never in excitation. And so that's how you get lack of disparity selectivity, because there's no correlation between the responses one filter and the other. And to sort of show you this with just a good example, here's a disparity selective cell and here's a disparity non-selective cell. And what that means is this information is being combined in left and right eye without being gated by disparity or anything. It's just mixed together. And that makes it really confusing what the neuron could actually be doing. However, this brings us to a hypothesis, which is actually what these nonlinearities do is they actually prevent mixing left and right eye information. And the reason is that if you actually add left and right eye before a nonlinearity, then you get these interaction terms in, for example, the binocular energy model and so on. If all you do is rectify in left and right eye without combining them, then while the response represents them, and the response will be inscrutable, you can imagine that these two packets of information are in the response, and different neurons acting like this will actually have them in the response to different degrees. And so I'll just make this is meant to be a quick point, which is that nonlinear mixing actually preserves other types of informations. So if you consider one of these neurons that nonlinearly mixes left and right eye information, what we did is we simulated a whole population of these and then presented a set of patterns to both the left and right eye at the same time. And these patterns are more like simulated where you have one pattern in one eye, like a binocular rivalry, another pattern in another eye. And then we hypothesized that because these things were nonlinearly mixed, that we could make a decoder that could then unmix them. In other words, you have a population of binocular neurons, but that you can actually decode the monocular information. And this essentially is true. And we just show that here with the color code. Again, the red cells are monocular. They can decode their eye of origin. The green cells are the ones that are binocular untuned, and they represent information. You can figure out what's in the left eye without worrying about the right eye or figuring out what's in the right eye without left eye, without actually, you can do them in the same sets of cells. You can decode what's in the left eye and right eye. And then, of course, the disparity selective cells, which mix these, linearly mix them, do poorly because they're really sensitive to the correlations between the eyes. And of course, what you can do is you can actually take a disparity 2 neuron that has these binocular receptive fields and convert them into monocular receptive fields and rescue this. So this is a general sort of strategy and how most binocular neurons might be important. But it involves this inscrutable nonlinear mixing of information, such that if you record from any one cell, it's going to have left and right eye receptive fields with nonlinearities and not mix them. So unfortunately, we didn't have the ability to go back. So this is at the point where it's a prediction. It'd be kind of neat to see because it's a pretty simple experiment. And yeah, and it's a strong prediction. So I just wanted to talk about what it's all mean. This is the 5 minutes of the talk that this built up to, and I call this sort of this idea is cortical dark matter, that it looks like from the perspective of disparity selectivity that V1 is a combination of kind of exquisite feature selectivity. And when you think about how you would need to have a convolution and the cells all at the same disparity and wire up these complex cells so that they're sensitive to disparity, that it really is this exquisite feature selectivity that we have the chance of really understanding at a circuit level. And the thing is this seems mixed with a big sea of what I call inscrutable computations, and we see them all the time. Neurons that are sensitive to both left and right eye information, but not selective to disparity. A lot of nonlinear cells that are mixing together things and don't have clear outputs. And there's really a question about why you would have this big sea of cells that don't have feature selectivity. And possibility one is I think maybe the canonical possibility that they actually are feature selective. We just didn't do the right experiment. And when you start talking about behavior, maybe there's behavioral contingencies that would make these light up, or just features that were not presented in the stimuli. But the other explanation, which I think is embraced by now increasing a lot of people, is that this kind of sea of dark matter is really parts of hidden layers that are not are stacked sets of nonlinear computations that are ultimately doing a larger purpose. And just one of the old ideas about that was that the sort of meaning in the natural world is not the same thing as meaning on the retina, and categories of different objects are going to be very hard to decode from retinal ganglion cells or early layers of visual cortex. And in that sense, the hierarchy is thought to apply possibly inscrutable nonlinear transforms that actually then untangle the representations of things that actually are behaviorally important to us. And if they're untangled, as this is showing, it then becomes straightforward to sort of decode them. In that sense, it's well known that linearly mixing different features actually makes it harder to decode arbitrary combinations of features, because if you have 3 features, but you linearly mix them, they're all in the same hyperplane, which means even if you have a whole lot of neurons, they're still in the same low dimensional subspace. In contrast, nonlinear mixing can project different features into a much higher dimensionality of the space, which can then be closer to the dimensionality of the number of neurons, or at least some fraction. And so it's thought that nonlinear mixing is actually extremely important to facilitate downstream decoding. In that sense, you might wonder why we would have feature detectors at all if you could just nonlinearly mix everything together and then easily decode it. And just really briefly, the idea is that V1 inputs actually have the highest resolution representation of visual information because they're getting the LGN inputs. And as a result, and it's also, there's a lot of spatial and temporal integration, including complex cells and so on, so that you add invariances to in fact, become selective to features at the expense of other things. But in doing so, you then get away from this raw output. So V1 might be a really great place to actually generate features in the context of this sea of nonlinearities and then package them up to be used later. And so the sort of picture the picture I want to say is that of a traffic light, and we can all recognize this as a traffic light without seeing colors at all. And presumably our whole object recognition system is doing this untangling through inscrutable computations going up. But if you wanted to know the color, it's much harder to get the color from nonlinearly entangled basic elements that were present at the level of the LGN. And so you might want to package up something like color in V1 and then just have that go ahead to the different areas where color would actually be important and play into whatever contingencies neurons are selected for there. And so just concluding, I feel like V1 is actually going to be this dichotomy. There's going to be parts of it which can understood, which might be using right strategies, might be decoded using kind of modern nonlinear processing. And this is the kind of stuff we can understand the purpose in algorithmic. What is frustrating is I think there's also a purpose to having inscrutable computations. And I'm not, and in fact, we can demonstrate if we know what a purpose could be, that of course you can decode from real neurons to get them, or in my case, decoding from a simulation. And so I'll leave you with this really question is it possible to understand V1 in this sense and the visual cortex in general? I'll just acknowledge the two students that did this work, now both gone. Felix Barsh was the grad student that did a lot of the disparity work, and then Ethan came in and did the decoding. And of course, our collaborator, Bruce Cumming, for all the data. And I'll just say that you're going to have another visitor in a couple of weeks working on things. We're working on things together that combine sort of this feature selectivity in color with deep neural networks to study color vision of the fovea, where there is really possibly clear circuits that can be decoded. So we're working on that. So thank you for your attention, and questions. Yeah.
I remember a little bit of another talk I saw recently. I am very forgetful on the details, but it was a Bethge lab talk where they also compared energy model and like neural networks fitted to different neurons. I think of maybe V1, maybe MT, but like to motion stimuli. And one of the conclusions that I remember from that work is that like neural networks kind of overfitted, that they were a lot better than the energy model in describing the response of the neurons to the. I don't quite remember. I think that they compare like a naturalistic stimulus set with like scenes and flying chairs to a stimulus set with like gratings and more like psychophysically like. I don't remember which one was the training one and that the transfer didn't happen very well. But yeah, it reminds me a little bit of that, of like, and I wanted to ask you like whether you have tested these models on some like stimulus set with different statistics from the ones trained, and whether you could expect something similar to happen, where maybe the energy model like generalizes a little bit more across like types of stimulus sets.
Yeah, so Yeah, I'll say a couple things. So this is just, yeah, I guess there's not too many people on Zoom. So I won't summarize. Let me just, I'll jump in and say that, that first of all, this question of generalizability is very large, and that's one motivation to actually choose the right mathematical form. So you need fewer parameters, and that the mathematical form itself will then correctly generalize to a new situation. And if you're clever, you can do that well. Presumably, our neurons have a set of parameters that they can vary, and there's a huge number of anatomical and physiological constraints. And so the neurons find certain solutions given those constraints, but if we don't know those constraints directly, we can have something that needs a lot more parameters and then will overfit. So I think that that's actually part of the goal of getting the right mathematical forms and trying to understand what the strategies are in a sort of more algorithmic model. It's not clear, just to be direct, whether these models generalize outside the sort of stimulus. In other words, how far they generalize. But it's an important test, and what we're working on in the in less of a toy model and more of a full model is to actually test a natural context, natural colored context, where essentially there's nothing to generalize to. Yeah.
The receptive fields that you showed near the end of the talk that I think you referred to, there are complex cell reconstructions of receptive fields that I think you referred to as inscrutable. These,
Yeah.
where you've got relatively strong weights in one eye and pretty close to zero weights in the other eye. We've been doing some normative modeling on the problem. Actually, Daniel has been on the problem of motion and depth. And what we've been finding is that you would actually want to have receptive fields with monocular selectivity in the eyes, so that it capture fast moving motion in 3 dimensions. The basic intuition is that if something's moving real quickly, you're not going to have corresponding image information within the receptive field in the 2 eyes, the binocular receptive field in the 2 eyes, simply because it moves through the receptive field so quickly that you want to be able to pick up monocular motion information. And I'm wondering if you've considered that scenario as one that might be useful for these types of receptive fields to help.
Yeah, God, yes. So actually, interocular, well, so the interocular velocity differences is actually one of the great examples that I know about, I think, where I think the pervasive models actually require monocular input, as you say, and this kind of problem would be easily solved in that sense, because even though the neurons are binocular, if you're doing any sort of decoding, you can extract monocular input just as easy as the binocular. I think that's part of it. So as to whether and so that does involve doing exactly this sort of nonlinear mixing. This is actually a pretty simple form of nonlinear mixing, and it's nice because it's like you have 2 eyes and they're sort of separate, even though the response of the neuron itself is kind of hard to understand. The underlying model isn't necessarily, but this sort of thing would hopefully make itself so that a decoder could use that sort of information. I'll be curious to talk to you about maybe how to make that. Yeah.
Following a little bit on these questions relating to disparity, computation, motion, computation. I mean, obviously there's a lot of overlap in what they're trying to do, but with motion, there's this sort of extra delay component. right? I mean, if you thought about what you'd expect if you have a system that's trying to do both and is trying to optimize, right? Like, so there's some subset is going to require extra delays, but they're all going to require some kind of correlation detection between, between the outputs of neurons at different points in space. Do you start to get these mixtures when you need to do both?
Yeah. So, gosh, there's two levels of that. So first of all, gosh, the second level just distracted me. Let me just think. The first level is one set of problems, which is probably I'll put to this side because I think you're asking about the second, which is that there's this conjoined problem of sort of knowing the distance of the object, as well as that it's the same image in both eyes. And in this sense, there's also a 3D velocity. And the interesting thing is because of the geometry, the 3D velocity is going to be different. And so there is, so I'll just put this aside, but there's a problem of solving all these things at once, which the delays can actually make it difficult. In other words, if you get object segmentation, will that feed back in time? The simpler aspect, which I think is an exciting thing about nonlinear mixing, is that I just showed a simple example, the one here, of nonlinear mixing for extracting just what the information is one eye. But you could probably just as easily decode a conjunction of two different velocities, one from the left eye, one from the right eye, in the same population of neurons, because it's mixing the velocities of the left and right eyes together in the nonlinear responses. So what nonlinear mixing does for you is it means that you can decode kind of complex contingencies, like between left and right eye input, kind of at moment to moment, I would say, just using the responses. Yeah. When I start thinking about like other features like color, there's a lot of where our perception of color is determined by the surround in very complex ways. And this is also something where you might want to non-linearly mix certain combinations of color and spatial information together so that you could decode kind of true color that's based on the context. But it doesn't have to do with the delays, which is a hard problem. Great. Any questions? All right. Well, thanks for your attention.